Good morning, my dear students. Today we will see the last topic of evolutionary relationship. So we will take one example of wild cabbage. You can see in this picture of wild cabbage. So the farmers done the selection, not natural selection, but artificial selection. According to their need, they selected and they breed and they form different types of plants. Okay, so wild cabbage only was there. Then they done so many changes for the plant and produce different types of vegetables. Okay, so we can see cabbage is a good example. So farmers over more than 2000 years cultivated wild cabbage as a food plant and generated different vegetables from it by selection. So this is artificial selection rather than natural selection. By our wish only we are selecting different types of we are getting different types of vegetable. Okay, so some farmers wanted to select very short distance between the leaf and they produce cabbage. Some wanted to select arrested flower development. So they breed, they got broccoli. And for sterile flowers, they made cauliflower. Some have selected for swollen parts and they got the vegetable known as kohlrabi. And some have simply looked for larger leaf and they got kale. So like that they have done selection and they got different types of vegetables. So some changes in the they also got red color cabbage. So leaves, only leaves that is known as the kale and the swollen stem that is known as the kohlrabi and the sterile flowers that is known as the broccoli. Sterile flowers they made cauliflower and the arrested flowers they made broccoli and the leaves that is not that they form cabbage. So from the wild cabbage different varieties of vegetables they made. Now let's see a video on this example of cabbage showing the evolutionary relationship.
There are also many characteristics to prove evolutionary relationships in plants. In the present scenario, farmers use plants with desirable characteristics to produce new varieties by a process called artificial selection. Farmers have descended all these vegetables from the wild cabbage ancestor. Short distances between leaves gave rise to a new variety called cabbage. Arrested flower development gave rise to a new vegetable, broccoli. Sterile flowers gave rise to the cauliflower. Swollen parts gave rise to kohlrabi. And larger leaves gave rise to a leafy vegetable, kale. These changes are observed due to changes in genes which accumulate over a period of time. The tools for tracing evolutionary relationships, like excavating, time dating, studying fossils, and determining DNA sequences, have been used for studying human evolution. All humans are a single species. The earliest members of the human species, Homo sapiens, are traced to Africa. A couple of 100,000 years ago, some of our ancestors left Africa and slowly spread across the planet. It resulted in stopping the gene flow, and over time it led to the evolution of different races. It is very crucial to understand certain things in tracing evolutionary relationships. Firstly, multiple branches are possible at each and every stage. For example, it is not true that human beings evolved from chimpanzees. Instead, both humans and chimpanzees had a common ancestor. Changes in DNA during reproduction are the basic events in evolution. Comparing the DNA of different species allows us to estimate how much the DNA has changed during the formation of these new species. In fact, there is no real progress in the idea of evolution. Evolution is simply the generation of diversity and shaping of the diversity by environmental selection. The only progressive trend in evolution seems to be that more and more complex body designs have emerged over time. However, many old and simple designs still survive. For example, one of the simpler forms, bacteria, is able to survive in almost all climatic conditions such as hot springs, deep sea thermal events, and in Antarctica. In other words, human beings are not the height of evolution, but simply yet another species in the crowded spectrum of evolving life.
Another way of tracing evolutionary relationships depends on the idea that changes in DNA during reproduction are the basic events in evolution. If that is the case, then comparing the DNA of different species should give us a direct estimate of how much DNA has changed during the formation of new species. So, this method is now extensively used to define the evolutionary relationship. So, some tools we have seen for the tracing of evolutionary relationship are excavating. That means check the fossils. Next one, time dating, how much age the fossils are, then studying the fossils and determining the DNA sequence. We can see great diversity of humans in planet. They are known as the races. So the different types of peoples that we can see around the world that we can call as races. Some are pale color like yellow, little cream color, then black, white or brown. So all humans, if we are taking human, we all are one species that is known as the Homo sapiens. Okay, so first when we search that excavating and all, first human fossils we got from Africa. So we are assuming that from Africa, slowly people migrated to other places and they adapted their own characteristics and slowly they changed and they formed the different races. So, the earliest member of human beings are known as the Homo sapiens. They are traced the genetic footprints in Africa. From there, they migrated slowly to different parts of the world. Like all other species on the planets, maybe by accidentally or some other variation, small changes happened and slowly evolution take place and they adapted to new place. So that like that different types of races came on the earth.